Hi everyone. In this video, we are going to understand about the importance of hydrogen bonds and also what is the hydrogen bond, how this hydrogen bond keeps the two strands together that we understand. Apart from that, we also understand the other chemical bonds like covalent bond, which is present between the sugars of the same strand. So how these covalent bonds are different from the hydrogen bond that we understand in this particular video. Okay, also we understand why that adenine cannot pair with the can only pair with the thymine. Why not it cannot pair with the for example a guanine, or why it cannot pair with the cytosine. What is the a problem that the adenine uh, uh, to form hydrogen bonds with the cytosine, that we will understand in this video. Okay, so first let us see first let us understand the different types of chemical bonds. So there are three types of chemical bonds present, we know that. So they are called covalent bonds, ionic bonds, and hydrogen bonds. So in covalent bonds, when atoms comes together, so when atoms come together and forms a molecule, so in covalent bond there the two atoms they share their electrons to form the chemical bond between the atoms. So whereas in case of the ionic bonds, where when two atoms come together, so one atom donates the electrons and other atom receives the electrons, so that they form an ionic bond. So in case of hydrogen bonds, so there the story is completely different. So to form that hydrogen bond, so that so to form this kind of hydrogen bond, see, so that one atom must be the hydrogen and the other atom must be an electronegative atom, like a nitrogen it can be a phosphorus, sorry, it can be oxygen, it can be sulfur. So such electronegative atoms must be present. So this hydrogen atom which is also there as a partner, so this must be linked to the other atom by the covalent bond. That is very important. So the hydrogen atom must be present, so this hydrogen atom must be present with the other atom in a molecule with a covalent bond. So the other atoms, they can be any electronegative atoms like nitrogen, oxygen, or a sulfur can be present. Okay, for example, when we see here, see the no oxygen is present here, and see the hydrogen is present on these two bases. So as I said, no, so to form the hydrogen bond, one must be electronegative atom. See, here is the oxygen is electronegative atom, more electronegative atom, because it has lone pair of electrons, so it is ready to form bonds with the other atoms. Okay, so whereas you see the hydrogen, this hydrogen is it has a covalent bond with the nitrogen here. So now we can expect the formation of the hydrogen bond between these two atoms, such as the oxygen and the hydrogen. Similarly, we can see here this hydrogen atom is there, and also the nitrogen is there. So this nitrogen is electronegative atom, this is a hydrogen atom is there. So between these two also we can expect an hydrogen bond. That's why between adenine and thymine we can expect two between adenine and thymine you can expect only two types of the two hydrogen bonds, not two types. So two hydrogen bonds are possible between the adenine and thymine. And we must understand that from here that the amino group, functional group, the amino group which is present on the sixth atom, it participates in the formation of the hydrogen bond. Also the functional groups which are present, it is on the fourth atom. So on the fourth carbon atom of the thymine, where the oxo group is present, so between these two functional group we can expect the hydrogen bond. Similarly, we can see the third atom, so where nitrogen is present, so nitrogen forms a covalent bond with this hydrogen. So this one, and also the other nitrogen atom which is there on the this particular on this what I say no on this adenine ring, see this in adenine, so base, so that forms the hydrogen bond here. Okay, and a very important thing that see here, the ninth atom of this purine ring forms, which is nitrogen atom, so it forms the glycosidic bond with the ribose sugar. Similarly, here you can see the first atom of this pyrimidine ring, that is a first atom is a nitrogen, it forms the glycosidic bond here. So in case of pyrimidines, the first atom forms the glycosidic bond with the sugars. In case of the pyrimidines, where the ninth atom forms the bond. So that is very important to understand here. And when you see the guanine and cytosine, so in guanine and cytosine, we know that okay, three hydrogen bonds are present. Let's see, the cytosine here has amino group. So the amino group of this cytosine, and the oxo group of this guanine. So the amino group of the cytosine and also the oxo group of the guanine, they forms one hydrogen bond here. Oxygen, as I said, no, it can form, it is electronegative atom, so the hydrogen which is covalently linked to the nitrogen, so these two form hydrogen bond here. And also when we look into the other hydrogen bonds we see here, uh, this one, okay, this is the third third atom, which is a third atom here, which is a sorry, yeah, third atom here, so fifth atom here, sorry, it is a first one, so second one, third one, yes, third nitrogen atom it is here. So this third nitrogen atom here, and also the nitrogen present is linked to the hydrogen atom, so covalently linked to the hydrogen atom. So this hydrogen atom and the nitrogen present on this purine ring, these two forms the hydrogen bond. Similarly, we can see here the second atom has the amino group, so fourth atom has the oxo group, and second atom has the amino group. We know that in guanine two types of functional groups are there, so both amino group and oxo group are there. So that's why we can see here, like see these two functional groups, participation of these two functional groups in the hydrogen bond. So again the oxo group, we can see here, so this oxo group and this amino group, these together they forms the hydrogen bond here. So three hydrogen bonds are expected between the guanine and cytosine. Now let's see why that adenine and cytosine, they both cannot form they don't form hydrogen bonds between them. So to understand that, we can see this is the amino functional group which is present on the sixth, which are present on sixth carbon atom, okay. um, See, this is a sixth carbon atom which has the amino group, okay. And similarly we can see this is the fourth atom which has the amino group. And see here, both are the are the only donors, so there is no acceptor, like no, there is no nitrogen atom or oxygen nitrogen atom or oxygen atom to with the lone pair of electrons to form the hydrogen bond between them. That's why the adenine cannot form hydrogen bond with the cytosine. Similarly, when we look here, so here also both are, see, have a lone pair of electrons. There is, that means both are these are the donors only. See, these both are the, sorry, acceptors only. So sorry, yeah, these two are the acceptors. So we have matching. So hydrogen bond can be expected when one donor and one acceptor is present, or one but it cannot be expected between an acceptor and acceptor, or a donor and donor. So it cannot be expected between acceptor or donor. So it can be expected only between the donor and acceptor. So that's why adenine cannot form hydrogen bonds with the cytosine in a DNA. Okay, this is how these hydrogen bonds are present. So whereas these phosphodiester bonds which are present between the sugars, so these phosphodiester bonds, they are covalent bonds. So these covalent bonds are very strong bonds. That's why all the biomolecules which are in polymer structure, so they contain the covalent bonds, more covalent bonds than the hydrogen bonds, because they form the backbone. So that is very important. And see, there are no connections, there are no bonds between the bases which are present one on the other in a DNA strand. So only forces which are present between these two bases, between these two bases are called van der Waals forces. These van der Waals forces, they keep the bases close to each other, with some type of a bond between them. So these are called base interactions also, or we can call them as van der Waals forces. So these are not so strong bonds, so they are very weak bonds than even the hydrogen bonds. So these hydrogen bonds, they keep the strands together, whereas this phosphodiester bond, they provide the backbone for the DNA strand of opposite strands, DNA of opposite strands. So this is about the structure of the hydrogen bond, and how hydrogen bond is formed between the two DNA strands, and also the phosphodiester bonds which are covalent bonds which are present between the sugars of the same strand. Thank you.